Hello students, this is Dr. Shikara again. In this video, we are going to focus on looking into what type of questions had come in biotechnology in 2019. So what type of preparation probably would be helpful, and how to deal with the questions and options is what I'm going to focus in this video. So definitely, I think biotechnology is taking more weightage in the science and technology questions as we are seeing it here. And then I also feel that the difficulty of the questions are also really high. Some questions, the first time talking to you about here, a question that is straightforward, easy, should not be really difficult at all. So here in this particular question, as you notice here, they talked about Cas9 protein. This is probably, we can say, one of the hot topic in UPSC, because CRISPR-Cas9 technology had come at least 3 times in the exams, either in the form of a direct question or as a form of one of the options. So last year they also asked about pairing it Cas9 with the wrong one that they asked about. So CRISPR-Cas9 system is something that we need to be familiar with. And also for designer baby, reason of designer baby controversy, the same technology, CRISPR-Cas9 technology has been used in this. So this question is straightforward. They asked what is Cas9 protein, that is often mentioned in the news. So you probably know it. CRISPR is the RNA component and Cas9 is the protein component, which acts like a molecular scissor. It cuts the targeted DNA molecule. So it is a molecular scissor used in gene editing. So this is clear. So you can definitely rule out anything else, like it is not a biosensor used in pathogen detection, and it is not a gene that makes plant pest resistance. Usually it is Bt gene that we talk about, and a herbicidal substance synthesized in genetically modified crops. So this is definitely something that is not correct. So it's molecular scissors. This is a straightforward answer. So something like this you should be able to answer without much difficulty, I feel. And next we'll talk about statements related to hepatitis B and HIV. As I was telling you in the last video, they are asking on comparison between different infectious agents. In this particular case, hepatitis B and HIV. So they are asking us to identify which of the statements is not correct. So you can first confidently identify the statements which you think are correct. Let's see. So hepatitis B virus is transmitted much like HIV. Do they follow the same path of transmission? Indeed yes. Why? Because both of them are sexually transmitted diseases. Either contaminated blood or sexual transmission is possible for both HIV and hepatitis B. I believe that this is something that you're probably familiar with. So you shouldn't have a problem in answering it. So second statement, it's a hepatitis B, unlike hepatitis C, does not have a vaccine. You may or may not be aware about it, so I'll come to this later. And then let's look into globally, the number of people infected with hepatitis B and C viruses are several times more than those infected with HIV. This you have to guess. And indeed, the thing is, if you read WHO website, there are over, I think, 325 million cases are reported with respect to both hepatitis B and hepatitis C. Okay, and both of these definitely around 9 times show more infections than HIV. This is the information that is given in WHO website, clearly comparing between hepatitis B and C and HIV. So sometimes standard sources like WHO website probably are the things that we have to read. Okay, just more than a newspaper sometimes related to these diseases. So definitely this is something that is correct. So now we have to choose, I think, something that you're not sure about, B or D. Some of those infected with hepatitis B and C viruses do not show the symptoms for many years. Meaning, do they stay latent? Do they stay actually inactive without causing a problem? Yes, this is a possibility with hepatitis B and hepatitis C. Okay, so this is also right. Then we are left with option B. Hepatitis B, unlike hepatitis C, doesn't have a vaccine. I'm sure you all heard about this. Like hepatitis A and B, we have vaccines. If you have read about Mission Indradhanush program, which was also asked in the previous year's exams, like hepatitis B, we have vaccines as part of Mission Indradhanush, as part of a universal immunization program. But as such, there is no vaccine for hepatitis C. Okay, so they just confused you with hepatitis B and C vaccines, and B is the wrong statement, and this is the correct answer. Okay, I feel this is moderate question. This is not too difficult, medium difficulty that you may experience with this. The next question also, I feel probably will have a little bit of moderate difficulty here. This question is to do with which of the following are the reasons for the occurrence of multi-drug resistance in microbial pathogens. A burning issue in India, really burning problem in India, because with respect to TB, we are suffering from multi-drug resistant TB, and it has been reported that India had highest number of drug resistant TBs in the world, unfortunately. So this is a very, very important topic, and let's see if you can understand these statements. And we are asked to identify the right statements. Okay, so genetic predisposition of some people. Are we genetically predisposed? Meaning, do we have any mutations or anything in our body that makes us more prone to antibiotic resistance? So probably it is not running, because this is something we are talking about with respect to the pathogen which is gaining resistance. So it is not something to do with what we have, what targets we have. So definitely genetic predisposition in people is not connected to the antimicrobial resistance or antibiotic resistance in pathogen. So this is something that you definitely can rule out, but you have to know about this. They are talking about host, they're not talking about the pathogen. So this is something you should be able to figure out. Then taking incorrect doses of antibiotics to cure diseases, definitely we know that when we don't take proper doses, you are giving enough opportunity for the pathogen to build resistance. So definitely this is a right statement. And then using antibiotics in livestock farming, if you have read about National Action Plan that Indian government came up in 2017, the using antibiotics in livestock farming is one big issue, and one of the major points here is to regulate this. Okay, so this is a right statement. And this is something you probably are not aware about, that multiple chronic diseases in some people. Multiple chronic diseases in some people, can they put people at risk for multi-drug resistance? Likely, because why? People who have multiple chronic diseases like diabetes, for example, or things related to blood pressure, so heart related problems, this will put your immune system in a lower pace, and it probably is weaker. We are more and more prone to infections. You may happen to take antibiotics frequently, and when you happen to take antibiotics frequently, there's a higher chance that you're becoming a breeding ground, okay, for developing antibiotic resistance in that. So I feel this 2, 3, 4 are the right statements. You can definitely rule out the point 1. So the possibility here is option D. So 2, 3, 4 of them are correct. So definitely you can rule out option A and option C when you are aware about statement 1. Then you might have confusion between B and D, and unfortunately that's how the statements are given to confuse you. But I go with the 2, 3, 4 option here is D. Okay, right. So let's move on to the next question. So that was definitely a medium level, which requires a little bit of understanding and analysis. I hope you will implement this type of information when you are reading it in other potential topics of 2020. Right. So next we'll talk about, with reference to the recent developments in science, which of the following statements is not correct. I really like this question because they're asking your overall understanding in biotechnology to see which of these statements are not correct. So first one is functional chromosomes can be created by joining segments of DNA taken from cells of different species. Can we take DNA from different species, combine them to form one functional chromosome? I have not heard about this being done. Okay, yes, synthetic chromosomes were made by Chinese scientists in East, but not by joining DNA segments coming from different species like that. So this I'll put on a hold, and let's see the other options. So pieces of artificial functional DNA can be created in laboratories. Indirectly asked, this is what I liked about this question. Can we artificially synthesize DNA in the laboratory? Is DNA synthesis possible? If you heard about Human Genome Project, right? Yes, they are planning to synthesize human DNA, human chromosomes. It is possible to synthesize DNA. This is the next thing that we are focusing on. So it is definitely correct. And options are statement C. A piece of DNA taken out from an animal cell, can be made to replicate outside a living cell in a laboratory. Can we replicate DNA outside the cell? This is what is PCR about, polymerase chain reaction is about. For TB, Indian government is recommending, they are establishing it. Recently it has been shown, one of the most sensitive methods that we developed using PCR. We are able to amplify DNA outside the cell. Clearly, this is an application that we know in biotechnology. Then cells taken out from plants and animals can be made to undergo cell division in the laboratory petri dishes. What are we talking about here? Can we multiply cells outside the body? Yes, we can. This is tissue culture. They're talking about lab meat, clean meat. This is all about this. So definitely these three are correct. There's no doubt about it. So functional chromosome fusion is what is the right statement, because it is not correct. So this is the right answer. So option A is the right answer. So this requires a good analysis of what's been happening in biotechnology. So such questions have come, so directly remembering them, knowing them is not enough. Being able to analyze them is most important here. Okay, moving on to the last question that I would like to discuss in this video. This is the most complex one. So this is about RNA interference. RNA interference technology, something that I usually avoid talking to students or naming them, but I do talk about the application of it, because in biotechnology, when I teach about GM crops, genetically modified crops, especially when we talk about flavor saver tomatoes, this is a technology that is used in delaying the ripening in the tomatoes. But let's see if this RNA interference technology has anything to do. First of all, I would like you to know RNA interference means we are introducing a recombinant DNA into the cells so that the recombinant DNA, so where there is a gene, its product is complementary to the mRNA of your target, not the protein but mRNA of your target. So meaning it can bind to mRNA, and in the process mRNA gets degraded by the cell. So you are reducing the amount of mRNA that is produced by RNA interference. That is why it is called interference technology. You are interfering the process of protein synthesis. And then look at the statements and why this has gained popularity. So it is used in developing gene silencing. Can you silence genes using this? If you're targeting mRNA, if you target completely, yes, it can silence the gene product. So yes, this is the right statement. And it can be used in developing therapies for the treatment of cancer. I know this is a little bit of stretch, that we have to understand that yes, in case of cancers, some genes are overexpressed. So it is very important that if you use RNA interference technology, you develop a technology, you can stop those overexpressed genes. We refer to them as oncogenes. Yes, it is really useful in treatments of cancer. The third statement, it can be used to develop hormone replacement therapy. This is something that I would suspect, because hormone replacement therapy is what we do. We actually give hormonal injections or supplements that we have with respect to hormones. We don't have to control it at the DNA level, because they can go either way. So it is definitely not a technology that is suitable for dealing with hormones. The fourth statement, it can be used to produce crop plants that are resistant to viral pathogens. Yes. If you want to stop viral pathogens from growing, yes, you can interfere, because some viruses actually have RNA as the genetic material. So RNA targeting would definitely use so. Okay, so I would think that 1, 2, 4 are correct. So let's look if there's an option for that. Yes. So statement A is the correct one, and you definitely see that most of the answers had 1 as the answer, because gene silencing is an alternate word for RNA interference. So even based on that, you can guess it, and as you rule out hormonal therapies, so that is statement 3, you can definitely rule out these two possibilities. Correct? We are left with 1, 2 and 4. So defining factor is 2. So you need to know the connection that cancer can happen because of oncogenes. Right? Hope you understood how to deal with these questions here. I'll come up with more questions in the other areas of science and technology. Otherwise, I will say goodbye for now. Thank you.